This is One-on-One. Dr. Reginald Farrow is Research Professor of Physics at the New Jersey Institute of Technology and JIT. Good to see you, Doctor.
It's great to be here, Steve.
You know, in preparation for this interview, I was saying, you're trying to create the world's smallest biofuel cell. And you said, yeah, but that's not exactly it. And I'm saying, no, I'm going to ask you what exactly you're trying to do. So go ahead.
Well, what I'm trying to do is make the world's smallest electrical probe. And the best way to get an idea of why I would want to do that, the cells in your body are like little bodies in their own right. I mean, they have a skeleton, they have function,
Hold on, back up. The cell's a body?
The cells inside your body. Yes.
Okay, go ahead.
So if you wanted to study your body, you'd want something smaller than that in order to figure out what's going on. Well, I needed something smaller than the cells in your body to study them. And then the question became, well, how do you make something like that?
Right.
And so I came up with a way to make a very, very tiny probe. And along the way, I figured out ways to make it functional, to make it be able to detect not just electricity, but be able to detect interesting things like glucose, and that's where the biofuel cell
What do you mean a
came in.
probe?
It's just a little tiny wire. That's really all it is.
Okay, so show us, because you brought something in.
This is the whole thing, but the actual part that's interesting about it, I could fit 7 of them inside your human hair, inside a diameter of a human hair.
Say what?
I could fit around 7 of them inside the diameter of a human hair, yes. So it's, and then the probe itself is even smaller than that. It is 100 times smaller than that.
And tell us from a layperson's perspective, if people are trying to understand the value, the benefit of this, give us a sense of just a couple of the specific benefits that would be derived.
Well,
could be derived.
The best example that I could give is, you know, there are a lot of times when you want to know what type of cell is there, like is it a normal cell, or is it healthy, is it sick, is it a cancer cell? There's some cases where you'd want to know, well, how are the cells getting along with each other?
What do you mean?
They communicate. And so you'd like to know, well, how well are they communicating? How are they sending signals back and forth?
Well, let me deal specifically with a disease. Say you're talking about cancer. What you're talking about, these incredibly small microscopic cells, what could they tell us?
Well, in the simplest way, in the simplest, application, they could just tell that the cancer cell is there.
Beyond that?
Beyond that, you could probably figure out how fast they're reproducing.
And that's beyond what we know right now.
It's something that we can do now in a way which is a lot slower than I'd like to be able to do it, and a lot of people would like to be able to do it.
How and why did you become fascinated with this?
It was one of the most interesting measurement problems that I knew of, and I came to this from being a physicist, not a biologist.
A physicist?
Yes.
Go ahead.
And it looked like one of those measurement problems that was out there that could use some state-of-the-art nanotechnology to solve. And so I actually left physics in the way I was studying, the way I was doing research before, to do physics and with a biological, concentration. And so I had to learn cell biology.
For those of us who have no idea what you're talking about, what are you talking about? I'm being serious. You go to a cocktail party and someone says, so how'd you get into this? And you say what you just said, and they go, but I don't get it. And they ask you to break that down with a for instance or for example. Meaning you were studying physics in a certain way.
Yes.
And that wasn't getting it done for you.
Yes.
And you wanted to take your background and do what with it?
I wanted to take my background and apply it to a very interesting measurement problem, a problem that could actually help people's health. It was the thing which actually turned me on was I saw that people were trying to make something called an artificial retina.
An artificial retina.
Yes.
That was the thing that you were into.
That was what got me involved in the first
Okay.
place. This is something to replace the retina in your eye. And I looked at that and realized that that would be something very difficult to do because you needed to make contact with the parts of your eye that actually go to the brain.
Wow.
And I said, now that's a hard problem, but I had an idea that I think I could offer something to that. And along the way, it just became a general interest in how do you make contact to cells? Because the cells are the things that communicate that information to the brain. And it eventually got to a point where I said, the problem is interesting, but then how do you make it? And it was beyond the technology that we had at the time, and so I had to develop new technology in order to realize what I wanted to do. And as in many cases in scientific research, we got lucky.
Well, I don't know about luck, but a team of people that you work with at NJIT, describe them. And what is a team of scientists like who come together on a project like this? And what is your conversation like? And could the rest of us be a part of it? I know it's a loaded question, but I'm realizing now that a group of people who are so smart and so smart in a particular way and communicate in such a unique and precise and specific way that the rest of us would feel lost. But it's a long-winded way of getting to this. What's it like being with these people?
Actually, NJIT is a very fertile place for doing research. The people there are very talented, and they're willing to talk to you. They're willing to talk about things which they know about, but they're willing to learn things that they don't necessarily know about. And that has allowed me to put together a group of people who have very diverse interests, because you have to be able to touch in a lot of areas in order to make something like I'm trying to make and study the things that we're trying to study. And what we try to do is speak in language that everybody can understand. And it's not just the people who are the scientists, it's the students themselves. And so we have to speak in a language that the students can understand too. And we have students ranging from high school up to people who have recently gotten their Ph.D s, and everybody needs to understand what they're doing. So we have to always break this down.
You're always breaking it down.
Yes.
So communication is a huge part of what you do.
Communication is everything about what we do, I would say. It's understanding, what's out there and being able to know. We have to communicate in order to know what to do.
You were born to do this work, weren't you?
Well, I kind of decided to do it when I was a teenager after seeing some work that was being done at Bell Labs. And I set a goal at that time to come to Bell Labs and work, and I did.
Well, I'm glad you're doing it.
Yeah.
And hopefully I'll get better at understanding it. Thank you, Doctor.
Well, thank
it's good
you.
stuff.
One-on-One with Steve Adubato has been a production of the Caucus Educational Corporation, celebrating 25 years of broadcast excellence. Funding for this edition of One-on-One with Steve Adubato has been provided by Wells Fargo, Horizon Blue Cross Blue Shield of New Jersey, the New Jersey Education Association, New Jersey Council of County Colleges, New Jersey Manufacturers Insurance Group, PSE&G, and by Verizon Communications. Promotional support provided by the Star Ledger and NJ.com, and by the New Jersey Business and Industry Association. Transportation provided by Airbrook Limousine. One-on-One with Steve Adubato has been produced in partnership with St. Joseph's Healthcare System.